end, Brianna. Okay, thank you. This is better. Okay, I'm very happy to now introduce our award winners for the Robert Cotter Prize, which is the prize for young investigators. And it is my pleasure to present our first person. This is Dr. Nicolas Young. He's an assistant professor at the Baylor College of Medicine since 2016. Dr. Young received his PhD in chemistry from the University of California, Davis, and with Professor Carlito Librilla. He received postdoctoral training at Princeton University in the Department of Molecular Biology with Professor Ben Garcia. He has also worked as the Director of Biological Applications at the National High Magnetic Field Laboratory with Professor Ellen Marshall as a staff scientist at Lawrence Livermore National Laboratory and in the biotechnology industry. Dr. Young uses proteomics to elucidate the mechanisms by which protein post-translational modifications modulate gene transcription and gene expression. In this endeavor, he has developed methods for the identification and quantification of extensive modified proteins by top and middle down proteomics. This work has involved the development of chromatographies that separate structural isomers of large peptides and intact proteins, mass spectrometry methods, data analysis algorithms for mixed tandem mass spectra, and methods for data interpretation to reveal mechanisms and biological function. His results have unveiled fundamental biological mechanisms and informed novel therapeutic routes for the treatment of diseases. So it is my pleasure to assign this award to Dr. Young, and we're all very much looking forward to your talk. Thank you.
All right, thank you very much. I'd like to start by expressing my appreciation for the US HUPO putting on this webinar and my deep appreciation for receiving the Robert Cotter Award. It's a great honor, and even more so to share it with my friend and colleague, Si Wu. So this audience, all right, slides are not advancing. There we go. All right, so there's no need to introduce the proteoform to this community. However, I want to emphasize that it truly is a necessary expansion of the central dogma, and that a protein-centric view of the proteome is fundamentally different than a proteoform-centric view of the proteome. So proteins can gain additional function through post-translational modification, can be turned on and turned off. But in the proteoform view of the proteome, it is the single molecule combinations on top of all of the other sources of variation that define the proteoform, and the proteoform is the bona fide, physiologically present single molecules upon which mechanisms can act and are the fundamental functional units that we look for in our hypotheses to understand how the biology functions. Nowhere is this more true than in the chromatin regulation system, which is heavily post-translationally modified on transcription factors, co-regulators, on the polymerases themselves that transcribe the genes. And it is this system that controls transcription and ultimately the translation into protein defining the proteome. And this is the center of what we study. So the building block of this is the nucleosome, at the center of which are histones. And these dense post-translational modification on the N-termini of the histones that extend out of the nucleosome license access to the underlying DNA sequence and control the transcription of these genes. And this system has writers that write PTMs, erasers that remove them, all in very site-specific manners. But interestingly, they have readers that can retrieve this information from this information-rich ledger. And this is through evolved domains that site-specifically recognize PTMs. But the most compelling part of this is that almost all of these readers are tandem readers. That is, they read more than one post-translational modification at a time. That is, that this entire system is a proteoform-based system. But yet, most of our knowledge of the chromatin regulatory system is through antibody-based methods that have really revolutionized our understanding of the non-coding or regulatory portions of the genome. Bottom-up proteomics has contributed greatly to this through the unbiased discovery of new PTM sites. But to get a full proteoform level understanding the mechanisms of the system, we need top-down or middle-down methods that are highly quantitative. And so people have worked on this since about 1882. A lot of work has gone in bottom-up proteomics in the early 2000s, Don Hunt, etc. However, looking at the proteoform level understanding of this starts with Neil Kelleher and Ben Garcia's work on doing middle-down proteomics using a Hillock-based offline chromatography. This was followed up by Josh Kuhn trying to take this online with a reverse phase chromatography, and he made an important contribution by making it clear that if you didn't achieve sufficient separation, it was a mathematical impossibility to deconvolve spectra with extensively mixed proteoforms. When I joined the Garcia lab in 2009, I developed an online weak cation exchange Hillock-based method that sufficiently chromatographically resolved these structural isomers and allowed deconvolution. I went on to join the Marshall lab, and in conjunction with Neil Kelleher, Liliana Pasatolik, and even Si Wu, and the Consortium for Top-Down Proteomics, we did an interlaboratory comparison that showed that there was actually relatively low concordance when there was not sufficient depth of coverage for all of the laboratories. We went on to develop methods for top-down proteomics on H2A, H2B, histone H1, and learned a lot of lessons in this process, such that differences in sequence can sometimes be misinterpreted as post-translational modifications, and that even very distal PTMs can have a strong effect on each other. And into what I'll talk about today is, in my laboratory, we've developed a rigorous, robust, high-throughput quantitative top-down proteomics platform for the biological study of the chromatin regulation system at the proteoform level. I should briefly mention that the proteoform problem is fairly distally related to the protein problem. A reverse sequence database really serves no purpose in this context, and protein or peptide level false discovery rates are not sufficient evidence for the claim of discovery of a proteoform. And this continues to be worked on by a variety of people, most notably led by the Consortium for Top-Down Proteomics. One of the problems here is that you can very quickly, if you combine all of the different known PTMs together, you very quickly reach trillions of possible proteoforms, more proteoforms for one given protein than there are proteins in the proteome. However, this is an information-rich system driven by biology. It has extensive hierarchies in it, and so not all proteoforms exist. But a major problem is that they are structural isomers. That is that they'll frequently co-elute. They'll definitely have the same mass. They will co-fragment, and you'll have mixed spectra. One of the solutions to this is to have a mixture model. We use a mixed integer linear optimization model to quantitatively deconvolute these spectra when there is sufficient information, even if that information is in just a few ions. But our preferred approach is to do innovative chromatographies that chromatographically separate these structural isomers from one another prior to the mass spectrometer. And we combine all of these innovations in methods that always start with multiple biological replicates, multiple technical triplicates, always interleaved to avoid batch and ordering effects. And so with these approaches, you can get single spectra that are extremely nice looking, extensive coverage, high confidence. However, as you go down to lower and lower abundance proteoforms, your sequence coverage decreases, and any metrics of your confidence identity also decrease. However, you are setting an upper limit on the abundance of that proteoform as long as you have sufficient depth of coverage and sufficient cadence of sampling. We validated these approaches with a lot of different approaches, such as null hypothesis testing, to show that our statistics our p-values are an accurate reflection of statistics, and that we have false discoveries at high abundance as frequently as we have them at low abundance. There is no bias in the abundance in terms of false discovery. We've also validated the accuracy of this, or the ground truth, using multiple approaches, including doing top-down proteomics and middle-down proteomics simultaneously in a single run, as well as comparing it to bottom-up proteomics. And for the most part, these agree very closely with few exceptions. So this proteoform quantitation approach allows us to get down to about 10 parts per million in abundance, with Pearson correlations comparable to those achieved by bottom-up proteomics. It is biologically robust, same sample, same result, sensitive down to a fraction of a millimeter of tissue, multiple replicates, and very high throughput relative to what has been done in the past. And it is robust. We analyze 24 hours a day, 7 days a week, for weeks at a time. And one metric we use here is that the cost of this is about 47 cents per proteoform, making it competitive with doing a simple Western blot for analysis of a PTM. So we're very interested in epigenetic therapies because they are low toxicity, have shown extraordinary efficacy for a variety of indications. However, the relationship between what is being inhibited and the clinical indication is poor due to a poor understanding of the fundamental biochemistry. One of the most common classes of these are the histone deacetylase inhibitors. And in looking at these, we see that they actually induce changes in the epigenome within minutes, but in a very proteoform specific manner, such that they're dependent on pre-existing post-translational modifications and are highly proteoform specific. And in fact, beyond just this one pre-existing PTM, there's specific hierarchies that then beg the question of what is the specificity of the enzyme? Is it specific to a protein, or is it specific to a proteoform? And we've indeed recapitulated this in vitro systems, where we can show that this same hierarchy is enabled simply by the active site of the enzyme. We've also looked at histone methyltransferases, such as SUV420 using an inhibitor called A196, which converts, SUV420 converts K20 monomethyl into K20 dimethyl. Yet some cells are resistant to this drug, while others are prone. However, at the proteoform level, it is the resistant cells that have the fastest and widest response. And if we look at this 15-minute time point, we can actually resolve the mechanism of resistance of that these K20 dimethyl containing proteoforms are themselves acetylated through the activation of histone acetyltransferases that then block the demethylase activity to achieve the change that is expected. And with this proteoform level understanding of the fundamental mechanisms in hand, we can propose combinatorial therapies that shut down this compensatory mechanism with histone acetyltransferase inhibitors, and thus overcome the resistance. We're very interested in taking this into in vivo systems, and this is a mouse model of acute myeloid leukemia. And in this case, we see in the bone marrow of these mice, these highly acetylated proteoforms, and these are different than the ones we were just looking at. They are very similar in which PTMs are present, but they're in a different order. They're a different proteoform. And these proteoforms are known to be read by a proteoform reader called BRD4. And when we inhibit BRD4, in addition to AMPK, which is a common approach, we can see that we actually get extended mean-free survival in these mice where neither of these drugs acts as a monotherapy. This led us to start asking all sorts of questions about the in vivo distribution of histone proteoforms and what we can learn in the in vivo systems. And one of our surprises here is that we could separate the cerebellum from the cortex in a PCA analysis. And this led us to propose the brain histone proteoform atlas. And so using multiple mice at 2 months of age, we've dissected the brain into 11 different sections and done extensive proteoform analysis on each. Counter to our expectations of our collaborators, there's actually great changes or differences spatially in all of these individual proteoforms, and many of them are highly statistically significant. For example, the olfactory bulb, the hypothalamus, and the midbrain are high in transcriptional silencing signals. Whereas the cerebellum and the cortex are not, and this is specifically a very deep heterochromatin sustained silencing of genes. But the cortex and the cerebellum are high in these sorts of transcriptional activation signals, these hyperacetylated proteoforms. And there are even some real surprises in specific post-translational modifications that are typically very, very, very low in almost all tissues and cells that we've looked at, such as H4R3 monomethylation, are extremely high exclusively in the dorsal midbrain. And this is known to be involved in nuclear hormone receptor mediated activation of gene transcription. But one of the directions that we would like to take this is in understanding aging and age-related diseases, such as Alzheimer's disease. And so we've done a 2-month, 12-month, and 25-month study of the mouse brain. And we can see that most of our proteoforms are actually totally flat. However, some of them, and specifically K27 dimethylation, and K36 dimethylation, in combination with each other on the single molecule level, increased dramatically from less than 0.5% all the way up to 7%. And this is in our proteoform brain atlas. This is specifically located at the cerebellum, at least at 2 months of age. And so in conclusion, we're reporting the first mouse brain histone proteoform atlas. This is being prepared for publication. And we've discovered age-associated proteoforms in the mouse brain. We need to localize these at older ages. We have a number of samples that we're hoping to get through for this, such as extended health span phenotypes. We have mice that live 30% longer, as well as drug addiction models in rats and postmortem tissue in humans. And with that, I'd like to acknowledge my group, specifically Matt Holt and Tao Wang, that did much of this work, as well as all of my collaborators. And thank you all for attending and for the Robert Cotter Award. Thank you very much. Happy to answer any questions.
Right. Well, Nick, this was such a wonderful talk. I have to say, I have to also say PTMs are very dear to my own heart. So,
of course, yes.
Certainly wonderful work. And I'm actually really impressed by all the many proteoforms that you're able to identify. Could you comment briefly on challenges for bioinformatics in this context also?
Yes, so the bioinformatics of this is quite difficult, not just in the primary data analysis, but also in interpreting the data and looking for these specific combinations, because there are so many different levels that you can look at. The full proteoform is really where we would like to see the most change, but we're also looking for themes and binary combinations, ternary combinations, and sort of dissecting it down in sort of a reductionist manner to find to figure out the mechanism essentially. In terms of the primary data analysis, we have built over the course of more than a decade a platform for interpretation of this data. We oftentimes compare our results to other data analysis platforms as well for validation.
Thank you. Yeah, and we have a couple of questions here from the audience. Simon Sidoli, he's asking, great talk, Nick. Do you observe modifications with still unclear biological function to be on hypermodified histones? It could be we still do not know their role because it is contingent to their partners, which we cannot analyze with antibody-based techniques.
Absolutely, and I think that so we frequently, function has been ascribed to certain post-translational modifications. I mean, Simone's asking about where function hasn't been ascribed, but frequently function is ascribed, but then it's really only in specific contexts on specific proteoforms that is having that function, and a lot of the post-translational modifications that have been used most extensively and have the most sort of injective one-to-one relationship with function are typically the ones at the end of hierarchies, where it's the last point in that hierarchy, and there are all of these post-translational modifications that may be necessary but not sufficient. And so I think there are, not just in the place where there's no function ascribed, but even where there is function ascribed, there's a lot more nuance and multiple functions that can be assigned. And when there are papers that are contradictory of each other, oftentimes they could both be correct, but there's some component that they're not observing because they're not looking at the proteoform level.
Sure. I have two more questions and then we'll end. From Iliana Christea, great that you can show modifications specifically enriched in different brain regions. This can be an important investigation in neurodegenerative diseases. How are you dealing with the heterogeneity of different brain regions, for example, different cell types per brain region?
Yeah, so I'm not showing this data today, but we have actually isolated specific cell types from specific regions as well in collaboration with some of our collaborators. And so this is critically important that not just to get region specific, but cell type specific as well. And that's through the expression of handles for immunoprecipitation on the surface of the nuclear envelope. And so we can isolate nuclei from specific cell types, from specific regions. And that's where we're going to gain even more insight. And we are working on Alzheimer's disease models as well for neurodegenerative diseases.
Great. Well, thank you, Nick. This was really wonderful. You will see there are a couple of additional questions for you in the question and answer section. Okay. So you could answer those. Thanks again. Very inspiring. Congratulations.
Thank you very much. Look forward to C's talk. Bye.
Great. So I would love to now move on to our next awardee for the Robert Cotter Young Investigator Award. Congratulations to Dr. Si Wu. Dr. Wu received her Bachelor of Science in Chemistry at the Anhui University and her PhD in Analytical Chemistry at the Washington State University with Professor James Bruce. She conducted her postdoctoral research on top-down proteomics at the Pacific Northwest National Laboratory, PNNL, with Drs. Liliana Pazzatoli and Richard Smith. Later, she worked as a research scientist at Battelle Toxicology Northwest and as senior scientist at PNNL. In 2015, she joined the Department of Chemistry and Biochemistry at the University of Oklahoma as an assistant professor. Her research interest focuses on developing and applying high-throughput quantitative top-down proteomics techniques for addressing important clinical and biological questions. So I'm honored to have you here talk about your science today. I think it is very important for you as HUPO to really express our congratulations to young scientists. So we're looking forward to your talk.
Thank you. Thank you, Brigitte, for the very nice introduction. First, I would like